is the Fitsug-Nagumo model. So I give you this as an example so that you can understand what we can do with such system. Probably you have heard about the Fitsug-Nagumo system because in neuroscience it's quite famous. So we have just two state variables, real state variables, great X, great Y, and dot X is equal to minus Y plus G of X, where G is a cubic function, while the Y dynamics is just the slow dynamics on Y is given by something that is linear in X and Y. Okay. And so the boundary layer system that we obtain is directly obtained by X dot equal minus Y plus G of X. Okay. So, yeah, exactly. The boundary layer system. And dot Y is equal to 0. And as you see here, we can analyze the stability of the singular point given by dot Y is equal to 0, meaning Y equal G of X. So this is the cubic here that you see. And you have, from anywhere where you start, for any initial condition, you reach asymptotically a certain attractive singular point. On the other hand, if you consider that you have the rescaling system, the rescale system that I presented earlier, we have the critical system that is only defined on the critical manifold G equal Y equal G of X, so the same critical set here, and we have the dynamics on Y on this critical manifold gives you the arrows, so the direction of the flow on this manifold. And to understand what happens for the whole system for epsilon small but not equal to 0, we apply geometric singular perturbation theory, meaning the persistence of what we call a compact normally hyperbolic invariant manifolds for small epsilon. It's a great, it's a complex sentence just to say that if we don't have any singularities of the singular system, we have a compact manifold here that is called a normally hyperbolic invariant manifold. And this manifold, it persists for small epsilon. So here, the three branches of the slow manifold here, of the critical manifold, sorry. Sorry. Come back here. The three branch of the critical manifold here persists into attractive manifolds very close to the critical manifold. That is the branches that are in orange here, and they are attractive, and the repulsive one, which is the red one. But we have some problems for the singularities in the fold of the critical manifold here. Okay, so we can control the asymptotic, but up to a certain point. What we do to understand what's happening to the flow near the critical manifold is a desingularization technique named the blow-up technique, which allows us to understand what are the invariants and first portrait of the desingularized vector field. We blow down and we do some local transition analysis, and we are able to decompose the return map and the whole and to make the whole global analysis. Okay, so it's not. I won't tell more about the underlying methods of analyzing the slow fast system, and oh, just this, which is the main strange behavior that we can see in such slow fast system, named the canard phenomenon. So mainly we have some great limit cycles that appears from this in the Fitsug-Nagumo system, the Fitsug-Nagumo model. But what we can have, depending on a certain parameter, we don't care if it's B0, B1, and B2 or B2. We can produce such phenomenon by any of these parameters. By changing very slowly one of the parameters, we can obtain such limit cycles that follows the repulsive manifold, the repulsive central manifold, during a while. And this is what we call a canal limit cycle, starting, and this is the transition between the completely stationary state and the appearance of a limit cycle, a great relaxation limit cycle. Okay. So let me go now to the models that I want to present today, which deals with the intracellular calcium concentrations in neurons. And I will take two examples for the network models, but I will mainly focus on the motoneuron model. But historically, I have developed the slow fast dynamics for a single neuron in another context than motoneuron. So the level of ICC in neurons, it is directly driven by electrical activity through a mechanism that is called calcium-induced calcium release mechanism. And what is quite simple with ICC is that experimental data can be obtained using fluorescence techniques. And from that, we recover some ICC time series. And I think that the first ones, the first application of that was on the GnRH neurons. So GnRH neurons, GnRH is a gonadotropin releasing hormone. It's a master hormone for the hypothalamus pituitary gonad axis, and it plays a key role in controlling all the cyclicity in female mammals. And it's very difficult to obtain some experimental data on GnRH neurons because of the position, the situation of the hypothalamus. So people produce the following biological models from olfactory placards taken from embryos of rhesus monkey. And it has the property to in vitro build a certain network that is quite similar to the GnRH neuron networks that we obtain in grown-up mammals in the hypothalamus. Okay, and here is some snapshots. So here the numbers that you see are taken for seconds, the seconds, and we have the neurons and the fluorescence that indicates the level of ICC in each neurons. And that you see that it evolves a long time. More recently, motoneurons have been recorded. The ICC in motoneurons have been recorded in zebrafish. They stuck the head of the zebrafish in some kind of a jello, and they are able to locate all the motoneurons on the ipsi and contralateral side of the spinal cord, and to record the ICC along during a while. What is interesting is that mainly in the GnRH neurons or in motoneurons, the pattern of calcium oscillations, of ICC oscillations that we can record, are mainly the same. So either we have a great peak, a series of great peaks with a very regular frequency appearing, but with long, but with long quiescent space between peaks. Or we have something like periodic peaks, but with no quiescent space in between. Or isolated peaks with only some noise between the peaks and no regular frequency. We have these three types of time series of ICC, either in GnRH neurons and motoneurons. And at the scale of the network, that are in GnRH neurons and motoneurons completely different, we have, of course, different behavior. So in GnRH neurons network from olfactory placards, we have this amazing set of time series. So each color represents one single neuron. And as you see, you have some intermittent synchronization of the neurons, while between these two successive peaks of synchronization, we seem to have a network that is completely desynchronized. In motoneurons, of course, it's completely different, because what we obtain is some kind of clustering between ipsi and contralateral cluster of motoneurons. And what we obtain is an antiphase behavior between the two sides, which is quite natural to understand, and strong synchronization in each cluster between the peaks of ICC. Yet we have some kind of heterogeneity here. So in each cluster, we can have some neurons that spikes, while other neurons remain silent. Okay. And I was very interested in developing a compact model using the formalism of slow-fast dynamics that is well dedicated to reproduce the time series obtained in neuronal activity, either in the GNOH neuron networks and the motoneuron networks. So I developed a compact model of ICC dynamics, which is a slow-fast system. And it's quite simple because it is based on the Fitsug-Nagomo dynamics, but with a third additional variable standing for the concentration of ICC in one single neuron. So this is the system for a single neuron ICC dynamics. So it's an extension of the Fitsug-Nagomo, as I said. So the function f is an odd cubic function, as in the, so it is S-shaped, as in the Fitsug-Nagomo system. We have the fast variable that stands for the electrical activity, the slow recovery variable y, which stands for the ionic activity, and the slow ICC variable, Ca, here. And as you see, the free rise function of x is the calcium dynamics induced by the polarization of the neuron membrane, okay? And it is taken as a sigmoid function. As here you start to see the additional parameters that can be taken, meaning the amplitude, the threshold of the sigmoid function, lambda, and the stiffness of the sigmoid function, rho Ca, okay? And the threshold of activation for this function free rise given by x of. On the other hand, we have in the calcium-induced calcium release mechanism some kind of feedback of the calcium dynamics onto the electrical activity, of course, because it brings some equilibrium between for the gates, the ionic gates of the neurons. When the intracellular calcium concentration starts to fall, it gives some equilibrium between the two sides of the membrane of the neuron. And so we have a calcium-induced hyperpolarization of the neuron membrane given by this Michaelis-Menten function here, with additional parameter, once again, mu and CAD, that gives us the feedback on the electrical activity by the concentration of ICC, of intracellular calcium. Okay. At last, sorry, TOCA here modulates the decay rate of the calcium level to the baseline. So I choose the baseline to reproduce the experimental data. And it's the second term of the slow ICC dynamics. Okay. So this system is quite simple, in fact, for a single neuron, but it is able to reproduce all the behaviors that we have identified in the experimental data. For the analysis, for the slow-fast analysis of this model, so we have S0, the 2D S-shaped critical manifold of this system, taking after rescaling the time, we put epsilon is equal to 0, and so the critical manifold is given by y is equal to f of x minus phi 4 of Cn. But since we are in a 3D space, the critical manifold, it has exactly the same properties as in the Fitzhugh-Nagumo system, but it is a 2D S-shaped null line. Okay, and we have two folds, f plus and f minus. And so the slow-fast dissection, exactly what I explained on the Fitzhugh-Nagumo system, gives us the row approximation and allows us to understand what we obtain in the to reproduce the experimental time series according to the value of the parameter. So this type of time series are reproduced by this system when we are in the case of the existence of a limit cycle. It's almost exactly the same as in the Fitzhugh-Nagumo system, but it's in 3D. Okay, and so the model output reflects quite well the experimental data of ICCs that we are obtaining in the GNRH neurons as well as in the motoneurons. If we change a bit the parameter and we try to reproduce, we try to reproduce the isolated peaks of ICC, then we change a bit the certain parameters to obtain a singular attractive point, an equilibrium, globally attractive. And starting from a certain location in the phase space, we are able to reproduce a single oscillation and go back to a stable equilibrium. The most important thing, so this is an asymptotic steady state, and the most important behavior that we want to reproduce is the following one, meaning the periodic peaks of the periodic oscillation of ICC, but each peak being separated from the successor, the preceding one, by a long quiescent space. And it seems in this experimental time series, as you see, we can infer that we have some kind of small oscillation of ICC between the two peaks, close to the baseline. To reproduce that, we have this possibility to have a phase space with a singular point that is very close to one of the fold. Here, the lower fold, F minus. And the dynamical behaviors that we obtain. So I show the model output here, which is quite good to reproduce such type of ICC time series. But how do we reproduce the quiescent space between two peaks? It's a mechanism of mixed mode oscillation, meaning the alternation between relaxation oscillation, large oscillation, and very small oscillation close to the fold that you can see on the magnified pictures that I show you here. How does it work? In fact, in the 2D case, remember that we have the slow manifold that we know it persists for epsilon small but positive. But we have these fold points that need to be understood. The dynamics should be desingularized, and we need to connect all the different orbits on the left and on the right, meaning the transition between the slow motion and the fast motion. And we have this the connection between the repulsive and attractive slow manifold gives us the explanation of the canard explosion. that I showed you here. In the 3D case, we have a one fast and 3D case with one fast variable and two slow variables. We have exactly the same thing, but we have slow attractive and repulsive manifolds that are not only lines, curves, but 2D manifolds in a 3D space. And what happens close to the folded singularity here is a wrapping of the attractive and repulsive slow manifolds near the fold line. And you can imagine that if you are starting from here, you are entering the neighborhood of this wrapped attractive and repulsive manifold, the dynamics needs to oscillate around the folded singularities because those are invariant manifolds. You cannot cross them. And because of this wrapping between the repulsive and attractive manifolds, you generate the dynamic generates small oscillation and remains close to the folded singularity during a while before it undergoes this fast motion of the dynamics and generate a large oscillation. Okay. So the second step from this single 3D system that we have developed is the parameter estimation to calibrate the interpeak interval and the amplitude that are mostly the more important thing in the deciphering the ICC time series in a network. And so we have a proposition, theoretical propositions that I have summarized here. It's simply a characterization of the ranges for amplitude and quiescent phase duration, and the protocol to fit both according to prescribed values. So we are able to reproduce exactly what we want for the quiescent phase, as you see, starting from, let's say, a basic time series generated by the system. We are able to make the quiescent phase greater or shorter, meaning here in case B, we have shorter quiescent phase for the blue line, but we have longer quiescent phase for the green time series. It's the same for the amplitude of the peaks. In case C, we are able to fit the amplitude, taking larger amplitude for the green time series and smaller amplitude for the blue time series. And what the ability that we have to fit both according to prescribed values using only two parameters, mu and k, is dealing with this, what we call the sector of rotation. I have not defined what is a sector of rotation, but it's in some sense the number of small oscillations that the dynamics needs to generate before undergoing the fast dynamics and produce a large amplitude. So the sector of rotation and the return map, we are able to perform some implicit calculation and to obtain some implicit dependence on mu of the number of rotation, meaning the duration of the quiescent phase. And we have a weak impact of this parameter on amplitude, as you see from A to B. K, on the other hand, another parameter impacts both the amplitude and the quiescent phase. And the protocol gives us the ability to match the amplitude with K using an approximation of the integral form and match afterwards the IPI with mu by selecting the selector of rotation. So we are able to fit any experimental data that we have recorded in a single neuron using this simple compact system. So I will present the first network that we have developed. It was about the episodic synchronization of GNRH neurons. Just to recall what this is what I call the episodic synchronization. Let me just 1 minute. Here is the episodic synchronization of calcium oscillation. As you see, we have some kind of synchronization at t equal 20 and t equal 80, 85, and afterwards at 140. But meanwhile, the network is completely desynchronized, and it seems that the GNRH neurons does not communicate and does not synchronize. But how do we explain and how do we mimic such kind of episodic synchronization between of the network? So it will be better to do that. Okay. Here. Okay, was there. Over there. Sorry. How do we perform such? How do we mimic such episodic synchronization? First, we need to have desynchronized neurons, and the variability in the two parameters mu and k in just one neuron, one cell, allows us to reproduce the asynchronous patterns and the variability in peak and amplitudes that we identify in the experimental data. So this is a simulation for 3 cells. And as you see, if you choose to perform 50 cells, we have this desynchronized behavior of neurons. So this is what we obtain by taking just the equation in xg, yg, cag, g being the identifier of one neuron. And we take great N neurons if we consider that they are completely uncoupled and that we have some variability in parameter k, so it depends on g, and eta, it depends on g. Oh, it's on, I changed I changed the name of the parameter. It's exactly the same as mu. Then we are able to reproduce this, but we introduce a global variable that is a representative of the global state of the network. Meaning that, in fact, since GNRH is secreted by GNRH neurons, we can understand that sigma is some kind of, it represents, in fact, the concentration of GNRH close to the network. And this global variable is given by a very, very slow, because GNRH neurons produce slowly GNRH, a very slow, so a third time scale in the dynamics, given by delta and sigma here, that are that are assumed to be small, positive but small, and a thresholded effect on each cell given by sigma sin. Oh, sorry, sigma sin is given here. Okay, sigma sin is a thresholded effect on each cell on the network, given by this global variable sigma. And once again, we choose a sigmoidal function to reproduce this thresholded effect. It's the most natural way to reproduce a threshold effect. And the reset mechanism of the variable sigma is induced by the episode of calcium synchronization, because if the calcium synchronization is not high in all the cells of the network, then it cannot overcome the parameter CRDC. So it's almost is equal to 0 unless all the neurons synchronize and the peaks of ICC synchronize among all the network. What we obtain is the following simulation. So just one thing about this experimental data. The gradual decrease of the signal is not something that we expect in the nature. It's just caused by the photobleaching, meaning the decrease of the fluorescence in the neurons. So the baseline tends to increase, and the amplitude of the ICC obtained through fluorescence imaging tends to decrease. Okay. So we can reproduce asynchronous phase using the variability in the entropic interval and amplitude, and episodes of unsynchronization with higher individual calcium peaks, a stable frequency, and even the post-excitatory suppressions that we see after the episodes of synchronization. And this is generated using a network model with 50 cells and heterogeneity just in K and mu. Okay. So this was the first network model that I have developed with people at INRIA on the model of neuroendocrinology network. And afterwards, with Soledad, we were interested in reproducing the ICC in motoneurons, which is much more interesting in some sense because the type of synchronization that we need to analyze is way more difficult to, it is much richer. So the experimental data we started were the same experimental data that I have already shown here, where you have, so this is, okay, it's called motoneuron, but in fact it's the different time series for each neurons identified on the picture above, and the ICC time series along time during 250 seconds. And as you see, in the left and right cluster, the neurons are perfectly synchronized. But on the other hand, from the left, between the left and right cluster of motoneurons, you have perfect antiphase, meaning that you cannot have synchronization between neurons of peaks of ICC between neurons with one in the left cluster and the other one in the right cluster. So it's quite natural to introduce a network model using a coupling of the electrical activity, because the calcium-induced calcium release mechanism is induced by the electrical activity. But it's through a an ionic. It's a slow mechanism. So it's quite natural to introduce a network connectivity using a coupling from the electrical activity onto the ionic activity. It is consistent with the time scale of the CICR mechanism. We introduce some heterogeneity in Ki so that we can understand. And the connectivity matrix is given by CIG. And to reproduce the ability of the clustering of the whole network between the left and right side of the spinal cord, we assume that the connectivity matrix is given by positive coupling gain between neurons in the same cluster, while we have inhibition between neurons when they are in two different clusters. So we have a connectivity matrix that can be given by block with positive value on the two blocks, top left and bottom right blocks, and with negative value on the top right and left bottom blocks. Problematic. What happens with the MMOs that are able to reproduce the quiescent phase that we can see here mostly in this either in the left or right side? What happens to the MMO when we try to synchronize the different cells and the different systems generating ICC time series in each neurons? And so we tried to infer the connectivity from the experimental data with cross-correlation. So it gives us an undirected network or Granger causality. Then it gives us some directed network. And the problematic was to understand how do we reproduce during the simulation? Are we able to reproduce with the simulation and try to find the value of the parameter that can generate the experimental data using the network model? So direct use of connectivity matrix, it does not lead to the right qualitative behavior because we need this parameter estimation in the model first. So we performed a synchronization analysis. Considering a first approximation, it's quite natural in dynamical system analysis to start from quite a strong approximation and to infer and to enrich the behavior by considering more complicated conditions. So the first synchronization was only to say, okay, if we consider that the ICC in each cluster are perfectly synchronized, then we only need to understand what happens in two coupled ICC oscillators. So we start by considering just two single systems here, X1, Y1, CA1, X2, Y2, CA2. And for the first approximation, we use the symmetry of the coupling by considering that we have the same coupling gain between O1 onto O2 and O2 onto O1. Okay. And the properties in this studied case, it means that we have identical cells, symmetric linear coupling. And for C equal to 0, we choose the parameters so that we have generated MMOs, and so quiescent phase in the ICC. So for C equal to 0, here we have the X1, X2 variable versus time. And of course, we are interested in the generated patterns of intracellular calcium concentration, meaning CA1 and CA2 in each cluster, or each neurons, if you prefer. So the bidirectional conditions on each nuclei and some arguments from singular perturbation gives us the characterization of the main behaviors of the whole model and the representation according to the coupling. And we see that even in these very narrow conditions, meaning identical cells, symmetric coupling, and so on, we have a very rich panel of behaviors according to the synchronization features. So the main asymptotic behaviors are the following one. For C negative, we have two cells that inhibit each other. Out of the model range, let's say that we have oscillation death, so nobody oscillates. But it's something that is almost, we see that already in the experimental data, so it's not completely out of the model. We have the possibility to reproduce one oscillation in one cell, while the other one remains silent. We have antiphase synchronization. This is what we obtain between two clusters. Two neurons from different clusters cannot spike at the same time. In fact, we have perfect antiphase synchronization due to the symmetry of the system. And for C, strictly positive, we identify mixed-mode oscillations. With slightly delayed synchronization, you cannot see that perfectly, but there is a small phase shift, even in the asymptotics in CA1, between CA1 and CA2. While we can have perfectly in-phase synchronization, meaning that both oscillators spikes exactly at the same time. And the repartition is the following. Perhaps I will, I see that we have, I speak a lot, so perhaps I will go a bit fast on this part. So we consider a certain solution with, let's say, a random initial condition, but outside the case where both oscillators would be the same, exactly the same, and leading them to exactly the same behavior. So it's not that interesting. Here I just introduce the crossing section time is going from negative to positive. So it means when the oscillator O1 for the state variable x1 for oscillator O1 crosses 0 from negative value to positive value. And the same one for x2, so O2. Then we have computed the sequences of return times, meaning, in some sense, the time lapse between two successive large oscillations of one oscillator. So we have two oscillators and two sequence of time, Ti x1 and Ti x2, depending on C, because it's the parameters that we are interested in, and the repartition of the behavior according to C. We also are interested in the cross return value, meaning that what is the value of the other oscillator when the state variable, the electrical state variable of the other oscillator passes through 0 from negative to positive value. And at last, the shift sequence, meaning the time lapse between two peak of oscillation, but between O1 and O2. Okay. And now we are able to do some to perform the repartition of all this behavior A, B, C, D, E here according to parameter C, using what I have introduced just before: the return time of O1, the return time of O2, the phase shift, and the half return time between O2 and O1. And what we see is something quite complicated, quite simple, in fact, for C negative, but quite complicated for C positive. And let me show you what we obtained here using a video here. Okay, so we have C that is increasing, and we have x1 and x2 above. And here we pass through antiphase synchronization for the whole value in region C. Then we have almost in phase or perfect in phase synchronization of MMOs along the value for C equals 0 to almost 0.7. And afterwards, we have some kind of equilibrium, that stabilization of the oscillation that appears, where we have perfect in-phase synchronization between the two oscillators. Let me show you once again here, but only with snapshots. Here we have oscillation death. In sector B, that is very narrow, as you can see. Here we have one oscillation death, meaning that we have an oscillation for one of the oscillator, but not the other one. Here, I have computed here x1 and x2, but it would have been better to put CA1 or CA2. But it's, of course, if you have a large oscillation in x1, it means that you have a peak of ICC for O1. But you don't have a large peak for O2 in blue here. So this is one oscillator death. And in C, as you can see, we have antiphase synchronization, perfect antiphase synchronization, because the shift here, the half shift is exact, 2 times the shift is exactly equal to the period of the whole system. But we have an increasing frequency with C of peaks, both in O1 and O2, since they are in perfect antiphase. And then we pass through some kind of chaotic behavior, close to 0. And that is the effect of very strange cascade of bifurcations. When C crosses 0, so here we are close to 0 but negative, and then you obtain for C positive but close some kind of stabilization. But as you can see, we have a lot of points here for the return map of both oscillators, meaning that we don't have what we call a limit cycle, but a whole set of periodic orbits and chaotic behaviors, even if the sequence, the time series, the qualitative behavior for the time series is quite stable. And here you can see that we have some very erratic behaviors appearing up to a large value for C, leading to the stabilization. And we have classified, and we have a theorem, really a theorem, giving exactly the value of C for which we have the existence of a unique couple of stable equilibrium states. It is case A. In B, the existence of an antiphasic quasi-periodic orbit with relaxation of one single oscillator. So B is between A and B, but it's very and C, but it's very narrow. C, the existence of a perfect antiphasic limit cycle with relaxation of both oscillators. And in D, D is the existence of MMO quasi-periodic orbits and MMO limit cycles as well, meaning case D or E, perfect or almost in phase synchronization. And the existence of an attractive MMO limit cycle in case E here. So it is really for x equal x1, x1 equal x2, y1 equal y2, ca1 equal ca2, meaning that both oscillators act exactly the same. Okay, and we have also the approximation of the transition values using the bifurcation theory, and because all this transition corresponds to the bifurcations. So I won't go into details, but the transition between A and B is some kind of canard explosion, as I showed you earlier, due to the existence of a singular Hopf bifurcation, and we have an explicit approximation in the singular limit for the critical system. The transition between B and C is due to what we call a pitchfork bifurcation of singular points. We also have an explicit formula. And we have also a pitchfork bifurcation of limit cycle. But outside the theorem, both bifurcation for the same value of C, we don't know if it's right or wrong. This is something that is still an open problem. But the approximation, so here is the approximation of the values CD and C0, and what we have with the numerics, and we see here, so the plain line are the explicit approximation, and the diamonds are the numerics, and you see that we have an excellent approximation for both values CD and C0, corresponding to the transition from A to B and then B to C. C to D is the stabilization of an hyperplane, a 3D subspace, and we have an explicit upper boundary of this. And the upper boundary here, once again, we have the explicit approximation, explicit over approximation, upper boundary, and the numerics. And you see that it's not as good as the former explicit approximation, but it gives us some idea. And we also have a theorem explaining directly the frequency increase during the inhibitory case, meaning that inside the C region, we know that the derivative of T1 according to C is negative, as well as the derivative of T2, meaning the two phase shift, the two duration of quiescent phase in both oscillators are negative and decrease with C, which means that the frequency increases. The key point of the proof is are based on the implicit reduction to the slow manifold, as I explained at the very beginning. And the control of the error gives us an integral expression of the durations. Differentiation under the integral and balance between the terms in the derivative of T2. So it is a quantitative result based on a qualitative analysis and geometric singular perturbation theory that at the beginning are completely qualitative properties, but making extensive use of this GSPT, we are able to retrieve some quantitative results. And since it's a bit late, I will pass. Here is a magnified view of what happens close to zero, and which mainly is mainly something very difficult to understand. But as you see, you have some kind of chaotic behavior close to zero for very small value when the coupling gain is negative, but it remains very complicated for a large value, almost, let's say, medium values of the coupling parameter C when it's positive. In fact, it means that we have non-hyperbolicity, what I called singularity at the beginning of my speech. This is the non-hyperbolicity in the uncoupled case that gives us such kind of theoretic phenomenon. So we don't have any persistence results for the weak coupling. The weak inhibition gives us chaotic behaviors. The weak excitation cascade of period doubling leading to chaos. But we still have some MMOs, and we keep this quiescent space in the ICC. And this is a magnified view of what happens for very small values of C. No, it's not a good one. Okay, sorry. There is some kind of problem in my keynote speech. Sorry. Okay. Just give me 1 sec. I'm sorry. It seems that the simulation was too big for for Zoom. Okay, I come back there. Okay. And afterwards, what we have done is the extension to weak heterogeneity of our analysis. When we have weak heterogeneity between cells, meaning that we don't have the symmetric case with O1 exactly the same as O2, let's say symmetric to O2. So we input some some heterogeneity between cells using different value of K1 for K. So K1 and K2 for O1 and O2. And the persistence of hyperbolic invariance allows us to retrieve the singular points, the limit cycle, the invariant subspace. So weak heterogeneity is very, very simple to understand. We know exactly what happens in all the gray area, the green area that we have here. Close to the transition between different behavior, it's much more complicated. But in here, we know what happens exactly. And we also have the persistence of generic bifurcation, what we call generic bifurcation, meaning hop, period doubling, and so on. So we can, in the case of weak heterogeneity, we have proved that almost the same behavior can happen. We have some holes in the theory, in the pink areas, because we don't know, we have unknown impact of the perturbation and no theoretical results yet. But the main repartition, even in the case of weak heterogeneity between cells, is acquired according to the coupling parameter. Okay, and we make use of this extension here. So we retrieve exactly, now we have the heterogeneity in the amplitude, in the quiescent space, and so on. And we will make use of this. So I skip that for okay. Sorry. I would like to come to go to the whole network. Okay, so now here's the protocol for parameter estimation for the small motoneuron networks. Okay, in this experimental analysis, we have 12 motoneurons. In the zebrafish, there is not more than I think it's 16 motoneurons. So the protocol to fit the mean frequency first fit the mean frequency and amplitude over clusters, meaning that we are able to have an idea of what should be K1 and mu1 and K2 and mu2, 1 for the left cluster, 2 for the right cluster. Starting from there, we fit the inhibitory coupling gain using our diagram of repartition of behaviors, and it gives us, since we have a precise, very precise analysis of the frequency of the frequency in the antiphase case, then we are able to know what is the coupling gain, the main coupling gain, the average coupling gain between clusters. So we are able to produce some strong antiphasic synchronization expected by the theorem, even if we consider exactly the same in each cluster, exactly the same value of the parameter. At the end, what we want is to have this heterogeneity. We leave it to the weak heterogeneity in the Ki. And what we obtain is something that represents quite well the qualitative behavior and even some kind of quantitative behavior in the motoneuron networks using our model with a few neurons. So we have a lot of conclusion and perspective. In fact, we have also developed, but I don't have time to explain. I had to do some choices to explain this work. We have developed also a reduced order method so that we are able to simulate quite efficiently a very large network of motoneurons, slow fast systems, because numerically it's quite difficult to capture all the dynamics in a slow fast system. But we have different perspectives. So first, the dynamical features. The first results on MMO and synchronization in two coupled oscillators. We want to extend this to a whole class of models, in fact. But the big issue is to characterize the local dynamics. We don't know exactly if what we have done on the MMOs on this system can be generalized. And this is the theoretical mathematical problem that we have. Something that is more of numerics, but also some theoretical problems. To decipher between period doubling cascades and other very intriguing types of transition that we obtain for C positive. And as you can see, we have, I put some colored dot here, and each value of C corresponds to a certain type of bifurcation between two, let's say, stable in-phase behavior. But all the bifurcation seems to be completely different. We have a period doubling cascade and inverse period doubling cascade, or it seems something that it's a pitchfork, but some kind of cascade of pitchfork bifurcation and so on. And it's very intriguing, and we are very interested in that. And once again, the variability between cells, which seems to be essential. And we would like more, even if our model is able to reproduce quite well the qualitative results obtained in the experimental data, we would like to involve back cross-correlation or Granger causality in the model fitting protocols so that we know exactly what will be the quantitative features of each neurons, and in that we are able to reproduce exactly the experimental data, at least for the zebrafish. So the exploration of the role of connectivity versus heterogeneity, it's something that we would like to perform using our new tool of our ROM model, our reduced order model, to obtain some fast simulation of large network. including the impact of eternal inputs, it's something that is not that difficult compared to what I have said just before. But for instance, periodic visual stimulus, it's something that has been done in experimental data. So they pass in front of the zebrafish that is stuck in his jello. They pass some dots, some black dots, and they have recorded the ICC time series in motoneurons in the zebrafish under this periodic visual stimulus. We would like to reproduce some kind of this kind of experimental data. That would be very interesting to understand how the network construction of the peripheral nerve. Thank you.
Thank you very much. So thank you very much, Alexandre, for this presentation. I think that we have a lot of questions. And let me start with just a remark, a general remark, concerning the use of Fitzhugh-Nagumo model. My understanding of this model is that it comes like a qualitative representation of the model proposed by Hodgkin and Huxley. So, and Fitzhugh-Nagumo comes like a very simple model with this cubic polynomial representing the behavior of the dynamic of the motoneurons. So that has been said. You use this model at the motoneural level, then you move to a more general model at the network level. So I just would like to precise, okay, the scale of using this model. Is it when you talk about motoneuron, you talk about population of motoneurons?
In fact, Fitzhugh-Nagumo system, as well as Hodgkin-Huxley model, is really to represent the electrical activity in a single model, in a single one, not a population. And Hodgkin-Huxley is very good, and it's quite specific and precise in reproducing the electrical activity. But it's a bit complicated to analyze mathematically. On the other hand, what we can do with the Fitzhugh-Nagumo system, which is more compact, and also represent greatly the relaxation phenomenon between in the electrical activity. We can plug some, as we have done, some other state variable so that it represents greatly what we have, the approximation of the electrical activity, but retrieving some other state variable. But it's always at a single neuron scale. So in here, what is great with the zebrafish, it's a few neurons, okay, just a few neurons organized as networks. And for each neurons, we have a single cell model, which has been developed to reproduce the electrical activity of one single model, one single neuron. And so it's really on this mechanics is quite direct, right? Natural to represent the network as one single neuron coupled with the other one. So it's not a masked model; it's really a single cell model and a network of single cell model.
Yeah, I'm talking about this because a lot of people use the Fitzhugh-Nagumo model at the level of the fMRI.
Yeah, that's right.
So at this stage, we are using this model at the population level. So it's a population of, and
of course,
this is mostly, in this case, compared to the mass neuronal model that you know very well. So my question is, okay, your choice goes to Fitzhugh-Nagumo at the single neurons and not at the
let's
population neurons.
let's say I use a Fitsung-Nagumo model as a reduction of the sorry, I'm a bit Hodgkin-Huxley model
That's what I
for
was saying.
for the single cell.
Yes, for the single. Okay.
But since we are interested in the ICC and not the electrical activity itself, but just the impact of the CICR mechanism from the electrical activity onto the calcium activity, it's not a big deal to have such approximation, and it's mathematically tractable. This is the main point.
Yes. Okay. So that's one point. I would like to ask another question and stop because I have another series of questions. But this one is about the fact that at the single neurons, you need. I was surprised that not seeing, for instance, for the motoneurons one Fitzhugh-Nagumo model because you introduce the ICC like calcium modulation with slow dynamic, and why you have not introduced something like the interneurons because the electrical activity is let's say two population of neurons. So why not two Fitzhugh-Nagumo to deal with the electrical activity and having the ICC like slow dynamics? I'm mentioning this because we introduced
because
yes
because it was already done in another context. I did no, in fact. At the level of the calcium dynamics, it would be very interesting to introduce something more mechanistic between neuron, interneurons, and so on. It would, if the first thing to do before introducing interneuron, if we want to make this model more sophisticated. It would be to use an extensive description of the state variable and not the Fitzhugh-Nagumo model. Because otherwise, here the X variable is here to say, okay, our neuron is in excitatory state, okay, and afterwards we have some quiescent space. If we want to couple interneurons that would, in fact, inhibit the motoneuron, I would have some problem to plug it on X, just the electrical activity. In that case, we would have we should have something more more precise, more mechanistic for reproducing the electrical activity of the neuron. Otherwise, the coupling should be a bit tricky, let's say not very physiologic.
Okay, so maybe we can discuss at another time because your lab developed this coupling between Fitzhugh-Nagumo models, and few years ago we talked about it with your supervisor, and so
that's right.
And we talk at that moment to include the calcium modulation in the coupling. So that's why I'm back 10 years later talking about this model because you choose another way, which is fine. So let me maybe give the floor to other question from the okay. Sorry. So, any question from the audience?
I might have one if there is no.
Yeah, go ahead, Christophe.
Okay, thanks a lot, Alexandre, for this very nice talk. So it's going to be more generic questions because I'm not expert in those models myself, but so I will talk about the kind of data and the kind of dynamic model I know, which is epilepsy, and notably epileptic seizures. And I know there's been a lot of work, and I think experimental work, notably by Christophe Bernard in Marseille or Catherine Sheldon in New York, and also modeling by Victor Gessard, knowing that an epileptic seizure also evolves a little bit that way, knowing that you have something very fast, which is neuronal activity, neuronal discharge, and the level of neuronal connectivity, as well as the level of the level of neuron connectivity, as well as the calcium, is going slow. And this looks, for what I've heard from talks from Marseille, as a very important process to stop the seizure. Like this slow evolution goes to something where connectivity goes down and goes up again, and then the seizure will stop.
Are you referring to calcium waves that we can?
There are some calcium waves evolved, but yeah, it's really the fact that you have this kind of coupling between a fast activity and a very slow activity. So I'm not exactly expert in that. So I was wondering if that kind of modeling, or I'm not sure what, I think Victor tried to model that kind of behavior, but I'm not sure how was applied to those data.
Starting from the beginning, in the GNH network, it's very difficult to understand if we have something like that. It's quite typical, quite specific, the neuron technology. For the motoneurons, for now, we focused on the mathematical analysis of a network model, and our benchmark was to reproduce the experimental data in the zebrafish, so with very few neurons. It would be interesting to see if we can extend some analysis to much larger network, and then we can understand what is exactly the impact of calcium waves and fast reaction, as you say, it's fast coupling that breaks for just very rapidly, and what is the impact of calcium in such case. For now on, we are focusing on understanding what is the interaction between inhibition and excitation in a simple case of a few neurons, and then in the simple case of a lot of neurons. And afterwards, perhaps we can understand what happens in more complicated and more sophisticated ways. But that's an idea. Perhaps I will go back to what I've done Bernard in Marseille on that.
Some of the data, they are actually, of course, there are very large networks involved, like an all brain networks, as we know. But the data from Catherine Sheldon are from Utah away, so that they are still quite local, like in terms of behavior.
Okay.
Thank you very much.
Thank you.
Thank you, Christophe. Any other question from the audience? Yes.
Actually, I don't know if you can.
You can speak.
Yeah. Thank you very much, Alexandre, for your nice presentation. Actually, I have a question regarding to this model and its compatibility with medical imaging. Has it been tried to use that synchronization and antiphasic calcium activity as a feature for medical imaging, that it can be used for normal and abnormal anti-calcium for the disease?
So first, I think Habib can confirm perhaps, but I think that Fetsung Nagumo has already been used in neuroimaging for resolving the inverse problem of EEG or something like that. If I understand well your question, specifically the antiphasic synchronization between types of clusters of neuron could be indicators of some abnormality or some pathology but in which context do you mean? Because in the context of motoneurons, it seems to be, on the contrary, perfectly physiological. It means that the ipsi and contralateral should not inhibit each other. Otherwise, the reaction to a stimulus could be completely annihilated because both sides act in the same way. But I'm not sure to have completely understood your question about abnormalities or. So can you specify?
Actually, this calcium activity, which would be related to electrical activity of the neurons, especially motor neurons. So
okay.
I have some experience. I don't know that MMO is NMO. I don't know it's in French or is other term of disease.
MMO, it's mixed mode oscillation.
I see. So, for example, can it be used, for example, for demyelination around the axons of the neurons? Because it's very important in medical imaging when the disease, especially the neuron disease, is directly related to demyelination, and it's related to the calcium ions between the cell membrane.
Yeah,
so
some hope I have would be to use this whole study to identify the problem of synchronization in motoneurons that could be the results of a pathology. In fact, in such sense, it means that if we are not in anti-synchronization, in antiphase, in fact, between clusters that should be in antiphase, then we can identify the type of abnormalities, identify the parameters that leads to this type of abnormalities, and then identify where is the problem in the physiological system. But we are 2 steps from that, not 1 step, but 2 steps, because we need to complete the analysis in a more realistic way, meaning with a lot of heterogeneity in the parameter and connectivity matrix that we are sure they represent the physiological case. But I see what it's possible. At the end, it would be possible to do that, to identify which parameters could result in such abnormalities in the fluorescence time series. That's right.
Thank you for explanation. Thank you.
Thank you. Any other question?
Can I ask you a general question?
Yes, you can ask. Yeah, absolutely. So maybe you have to go close to the.
First of all, thank you so much for your nice and informative presentation. I just want to ask about a general question concerning bifurcation. Let me start with this one. When we increase the dimension of differential equation, bifurcation is not easy to go. I mean, determine the bifurcation of the system. And I've read some works you developed concerning neural mass model, and you use some computational methods to determine the bifurcation. And we know that mathematically there is a specific definition for bifurcation.
Oh yeah, sure.
Yes, and I just want to know that how we can rely on computational method for addressing the bifurcation of the system. I mean, because you know, maybe you argue that for some value there is a specific kind of behavior in the system, but maybe I can find a specific value, and for this value it diverge of what you argue about it. I don't know. I can.
Okay. There is a whole literature about reliable methods for tracking bifurcation. In fact, bifurcation, numerical bifurcation, for tracking numerically the bifurcation of singular points, it's not that easy. It does not depend on the dimension. It's more the codimension, meaning the number of parameters that you take into account to understand what type of bifurcation you have in the parameter space. And in fact, theoretically, we don't have a whole, even theoretically, we don't have a whole description of all the bifurcation of singular points in more than 2 dimensions and more than codimension 2. So there is a lot of work to do on that. But the bifurcations that have been theoretically classified, there is a whole literature of numerical methods to track them, to follow them in the parameter space, and more specifically for bifurcation of periodic orbits, bifurcation of limit cycles. That is called the arc length continuation method. It's not just an inference of the bifurcation. Theoretically, on the numerical analysis properties of the methods, they have been proven to be efficient. If that's your question.
Okay, thank you for the response. Any other question, guys? If you don't have, I have one again. I have a question about the model at the network level. So the model at the network level comes with, when we talk about the network, we need to define what you suggest, like a connectivity relationship between nodes of the network. And this connectivity, in your case, comes from this modulation of the calcium. Modulation of the calcium define the connectivity. That's right?
No, the connectivity, the connectivity between neurons is, in some sense, it's electrical. But the electrical activity, the electrical activity, it comes since they are the motoneurons are quite away from each other. So it's some kind of aggregation of different synaptic connections, but it's not a coupling of the intracellular calcium onto the electrical activity or so on. In fact, if I come back to the model here, let me show you that in the network. Where is my network? Sorry, my network. Okay, let's see. Just here for two neurons. As you see, you have a coupling of the electrical activity onto the ionic activity, but the intracellular calcium concentration does not interact between neurons. Okay. It's a coupling from X on Y. So it's the electrical activity of one neuron that impacts the ionic activity of the other one. So it seems to be just a synaptic connection.
It's a synaptic connection.
Yeah.
So I'm just referring to what we did in the network level for the brain, the work that we do together. And I am just wondering if the connectivity is something that need to be related to the electrical activity or the metabolic activity, or a fixed connectivity, because even in the zebrafish we can know, I mean, this anatomical connectivity between the motoneurons.
Yeah. Yeah.
So
For now, we are considering fixed connectivity matrix. So the network is fixed. If we want to involve some changes in the network, it's possible. In fact, the connectivity matrix CIG could evolve with time. But in that case, I guess it's reasonable to put that on an even slower time scale, even slower than the calcium dynamics. You know?
I need maybe to understand a little bit more how you manage this connectivity at the level of the network. But anyway,
Okay.
there is a lot of similarities with a mass neuron model, and this is my last question. And the model that we have, including the GABA and glutamate, which is something like similar. It's not similar to the calcium, but this is another phenomena that we can include and interact. In your case, the calcium does not interact with the electrical activity. That's what you see.
The calcium activity interact with the electrical activity in each cell, each node, and of course the electrical activity the neurons interact through electrical activity.
Yes.
So as you see, here is the network. So it's a coupling of the difference of potential between, so it's a synaptic connection, the difference of potential between two neurons, I and G, and all the neurons, but it's not it does not involve a difference between the intracellular calcium concentrations between neuron I and neuron G. It will not have any sense because it's intracellular calcium.
Yes.
Okay.
Okay, that's clear. Thank you very much. If you don't have any other question, guys, we are going to let Alexandre moving to something else because he is right now in France and we don't have exactly the same time, and the kids are waiting for him, I think. Okay. Okay.
Luna was saying that he can film as well.
Okay, so again, thank you very much, Alexandre, for this nice presentation and answering our question. And hope seeing you soon.
Yeah.
Yeah.
With pleasure.
Okay, thank you very much.
Thank you.
Bye.
Have a nice day.
Thanks.
Salut Adil.
Merci Alexandre, à très bientôt. Okay.